Welcome to the Discovering the Structure of DNA slideshow. In this slideshow, you will learn about the structure of DNA and you will learn about the discovery of DNA. What is DNA? DNA is also known as deoxyribonucleic acid. The deoxyribo portion of the word represents the sugar that is found in DNA. DNA holds all of the information in a cell. In a eukaryotic cell, it's going to be located in the cell's nucleus. In a prokaryotic cell, the DNA will be located in the nucleoid region. DNA also has a huge job. DNA's job is to tell a cell what proteins to make. We call that coding for proteins. So DNA codes for proteins that are essential to life. DNA is a nucleic acid. The monomer of a nucleic acid is a nucleotide. A nucleotide has three parts. It has a phosphate group. It has a sugar. In this case, the sugar is deoxyribose. It also has a nitrogenous or nitrogen base. If you look at the picture to the right, you will see how all three of these components are connected together. DNA has four nitrogenous or nitrogen bases. The first is thymine, which is abbreviated with the letter T. There's also adenine, which is abbreviated with the letter A. There's cytosine, which is abbreviated with the letter C, and guanine, which is abbreviated with the letter G. We like to call DNA a ladder. If you think of a ladder, it has sides and then it has rungs, which are the parts in the middle that you step on. The sides of the ladder in DNA are made of sugars and phosphates, and they alternate: a sugar, a phosphate, a sugar, a phosphate. Nitrogen bases, or my adenine, thymine, cytosine, and guanine, are going to pair together and make up the rungs, or the middle part of the ladder. This next bullet has a typo in it. The atoms within the two sides of the ladder are held together by strong covalent bonds. They're really hard to break. In the middle, the rungs are held together by weak hydrogen bonds between the nitrogen bases. DNA was discovered as part of a collaboration by several different scientists. The scientists that we're going to cover are Erwin Chargaff, Rosalind Franklin, James Watson, and Francis Crick. Rosalind Franklin is one of my favorite historical scientific figures. She worked with X-ray photography to help to try and find the structure of DNA. So she wanted to know what DNA actually looks like. She took a famous picture called Photo 51. I'm going to show you that in a moment, which revealed DNA structure. And this was in 1952. Rosalind Franklin died of cancer in 1958. We tend to think that that's due to her prolonged exposure to X-rays without any personal protective equipment. So it's actually pretty tragic that she died, and I'm going to tell you why in just a moment. So here's her famous Photo 51. Doesn't look like much to me, but she was so brilliant that she used this to defend and deduce the structure of DNA. Okay, so Watson and Crick are also pretty important historical figures when it comes to DNA. I'm sure you've heard of them before because they are actually credited with finding the structure of DNA in 1953. Only that's not really what happened. So James Watson got a peek at Rosalind Franklin's Photo 51, and he and Francis Crick used that photo with other evidence to help them describe the structure of DNA. What they said is that DNA is a double helix and the strands are held together by weak hydrogen bonds formed between the bases. Unfortunately, this paper was published and Rosalind Franklin didn't get any credit because she was already deceased at that point. It's pretty sad, and we're going to talk more about that in class. So make sure you get your notes complete for this. Remember that that is an assignment, and I will see you guys all tomorrow. Okay, guys, so as you know, DNA has two strands, and so shown up here at the top are the two strands of one DNA molecule. They give us in the directions, and you should know from your notes that A bonds with T and C bonds or pairs with G. So the two that are curved pair together if C has a curve and G has a curve, and the two that are straighter pair together. A is a straight letter made of straight lines, and so is T. So, as you see in this picture, anytime there's an A, on the opposite DNA strand, there's going to be a T. Anytime there is a C on the opposite DNA, or I'm sorry, a G, on the opposite DNA strand, there is a C. So we are going to replicate the same thing they did at the top here for number 1. And I'm going to work out number 1 and 2 with you. And the rest you're going to do as part of your work this week. So let's begin. I have the letter C. And C is going to pair with G. So I will write the letter G below the C. I have another C. It's going to pair with a G as well. Now I have a T. Think to yourself, what pairs with T? A. So I'm going to write an A there. I've got the letter C. So I'll write a G that pairs with C. I've got two T's. And so I will write two A's because A pairs with T. Now I have another T. This T will pair with an A. I have an A. This A will pair with a T. I've got a C. This C will pair with a G. I've got an A. That A will pair with a T. This C will pair with a G. The A will pair with a T. This C will pair with a G. Now I've got two G's. They will both pair with C's. So I will write C see. I have an A. That A will pair with a T. I've got two G's, and just like before, those G's will pair with C's. I've got a G. That G will pair with a C. A T. That T will pair with an A. I've got an A. That A will pair with a T. Almost done. I've got a C. That C will pair with a G. My G will pair with a C. My C will pair with a G. My T will pair with an A. My A will pair with a T. The T will pair with an A. I've got a T that pairs with an A. I've got a C that pairs with a G. A T that pairs with an A. Last set of three bases. I've got an A that pairs with a T that pairs with an A, and a G that pairs with a C. And then I have finished that whole line. So what I'm going to do for number 2 is I'm going to write the answers out, and then we'll come back and talk about it, okay? Here is the answer. T, C, because we're pairing with a G. Another T because we're pairing with an A. An A because we're pairing, a T because we're pairing with an A. A G because we're pairing with a C. A T because we're pairing with an A. An A because we're pairing with a T. I've got two T's because we're pairing with two A's. I've got an A because we are pairing with a T. My next letter is an A, so that gives me a T as its partner, and so on. Okay, there is the answer to the second line. If you get lost, always remember that the two with straight lines, A and T, pair together, and the two with curves, C and G, pair together. So good luck finishing the rest of this assignment. As always, if you have any questions, don't hesitate to ask me, and I look forward to seeing everyone in class. All right, bye guys.